The human brain has virtually zero energy reserve. To maintain continuous electrical signaling, neurons rely entirely on oxygen and glucose for aerobic metabolism. This diagram details the cellular and molecular events of acute brain injury. When that metabolic baseline is disrupted by an initial insult, whether from an ischemic stroke, a hemorrhage, a prolonged seizure, or blunt trauma, the cellular supply line is cut. Regardless of the macroscopic trigger, all of these events converge on a single biochemical choke point. A massive clinical brain injury is driven by a microscopic 10-step cascade that begins the exact second oxygen is lost. Without oxygen and glucose, the neuron's mitochondria immediately halt production of ATP. ATP is the primary energy currency of the cell. Because neurons depend heavily on aerobic metabolism, ATP levels plummet instantly. There are no backup reserves to draw from. This severe energy deficit does not instantly kill the cell. Instead, it arms a self-amplifying sequence of molecular destruction. The first casualty of the ATP crash is the sodium-potassium pump on the neuronal membrane. Without energy to drive the pump, the membrane depolarizes. Potassium freely leaks out into the extracellular space, while sodium rapidly rushes into the cell. The sudden influx of sodium changes the internal osmotic pressure. To balance the gradient, water rapidly follows the sodium, flooding the intracellular space. This causes cytotoxic edema. The cell physically swells, compromising the structural integrity of the neuron from the inside out. At the synaptic cleft, the initial membrane depolarization forces voltage-gated calcium channels to spring open. This triggers an excessive, uncontrolled release of glutamate from presynaptic vesicles. Glutamate is the brain's primary excitatory neurotransmitter. The flood of glutamate hyperactivates NMDA and AMPA receptors on the postsynaptic neuron. These are specialized receptor proteins that mediate electrical signals between cells. With these channels forced wide open, a massive wave of calcium floods into the postsynaptic neuron. Inside the cell, this calcium overload triggers catastrophic damage. It activates destructive proteases, lipases, and endonucleases, enzymes that aggressively tear apart the cytoskeleton, degrade the cell membrane, and shred the DNA. This lethal feedback loop is known as excitotoxicity. The brain's reliance on rapid neurotransmission makes it exceptionally vulnerable to self-destruction once the system loses regulation. Simultaneously, the failing calcium-overloaded mitochondria begin generating reactive oxygen and nitrogen species. These are highly unstable molecules that aggressively strip electrons from nearby cellular structures. The cell is saturated with toxic molecular byproducts, including superoxide, hydrogen peroxide, nitric oxide, and peroxide nitrite. These free radicals inflict severe physical damage. They cause lipid peroxidation, which tears apart cell membranes, induce DNA strand breaks, and degrade the blood-brain barrier. Meanwhile, the initial hypoxia forces the tissue into anaerobic metabolism, leading to an extreme buildup of excess lactate and hydrogen ions. This severe drop in pH disables cellular enzymes. It also causes nearby astrocytes, the brain's structural support cells, to fail at their crucial job of clearing the excess extracellular glutamate. Reactive oxygen species and severe acidosis form a secondary wave of damage. They chemically dismantle the cell's remaining survival mechanisms while directly amplifying the excitotoxic loop. The brain's resident immune cells, known as microglia, detect this damage. They sense necrotic signals and DAMPs, warning proteins released by dying neurons. The microglia release key inflammatory cytokines like IL-1 beta, IL-6, and TNF-alpha. These chemicals recruit circulating leukocytes from the bloodstream, driving widespread neuroinflammation. The body attempts to compensate for the injury. In ischemic strokes, collateral blood vessels open up. In settings of high pressure, the Cushing response triggers, a reflex that elevates systemic blood pressure to force blood into the brain. But the brain operates under a severe physical constraint. It is entirely enclosed within a rigid, unyielding bone structure. This creates the confinement paradox. In other tissues, vascular swelling and inflammation are necessary for healing. Inside the skull, they rapidly spike intracranial pressure, turning a vital compensatory response into a fatal event. This microscopic molecular cascade dictates exactly how damage manifests at the gross tissue level. In an ischemic stroke, the brain tissue divides into distinct functional zones. The core infarct is the central region. Here, severe and immediate ATP failure drives fast, irreversible tissue necrosis. Surrounding this dead core is the penumbra. This tissue is salvageable in the early hours, but remains highly vulnerable to secondary waves of excitotoxicity, free radicals, and edema. This contrasts with other injury types. A prolonged seizure depletes ATP through extreme energy demand, while a traumatic brain injury pairs mechanical tissue damage with secondary inflammatory swelling. The entire clinical race against time in acute neurology is about shutting down this molecular cascade before it consumes the salvageable penumbra. The final microscopic fate of these cells depends on their location. Neurons in the ischemic core undergo rapid necrosis, while those in the penumbra suffer delayed apoptosis, a slower process of programmed cellular breakdown. Long after acute inflammation subsides, a process called gliosis occurs. Astrocytes proliferate and form a permanent physical scar. The brain requires massive amounts of continuous energy to sustain its electrical excitability. That same highly tuned architecture guarantees its vulnerability. Because of this strict cellular design and rigid physical confinement, a localized neural insult will inevitably evolve into a predictable systemic cellular collapse unless medical intervention interrupts the cascade.